Hello students, we will discuss about the types of immune response today. Introduction. Immunity, the state of protection from infectious disease, has two major components: innate immunity, adaptive immunity. The innate immunity is less specific component of the immunity and provides the first line of defense against infection. The innate immunity is broad reactive, that is, uniform in all members of the same species. The adaptive immunity is more specific immunity and it comes into play only after it encounters an antigen. The adaptive immunity is more specific to the antigen and has remarkable memory. Innate immunity. The innate immunity gives the first line of defense against infection. It includes molecules and cellular mechanisms pre-deployed before an infection and poised to prevent or eliminate it. It usually lasts for a short time and it's over within few hours. The innate immunity can distinguish self and non-self pathogen, but they are not very specialized to distinguish small differences in foreign molecules. The innate immunity composed of four types of defensive barriers: anatomical barriers, physiological barriers, phagocytic or endocytic barriers, and inflammatory barriers. Anatomical barriers. Physical and anatomical barrier that tends to prevent the entry of pathogens are an organism's first line of defense against infection. The skin, the surface of the mucous membrane are included in this category because they are effective barrier to the entry of microorganisms. The skin provides a mechanical barrier that retards the entry of microbes. The pH of the skin is also maintained around 3 to 5, and this pH inhibits the growth of most microorganisms. The alimentary, respiratory, and urinogenital tracts are lined by mucous membranes which secrete viscous fluid called mucus that can entrap foreign microorganisms. A number of non-specific defense mechanisms also tends to prevent the entry of the microorganisms. For example, saliva and tears acts to wash away potential invaders and also contain antibacterial or antiviral substances. Physiological barriers. The physiological barrier that contributes to innate immunity includes temperature, pH, and various soluble and cell-associated molecules. Many species are not susceptible to certain disease simply because their normal body temperature inhibits growth of the pathogen. Chickens, for example, have innate immunity to anthrax because their high body temperature inhibits the growth of the bacteria. Gastric acidity is an innate physiological barrier to infection because very few ingested microorganisms can survive the low pH of the stomach contents. A variety of soluble factors contribute to innate immunity. Some of them are the soluble proteins, lysozyme, interferon, and complement. Lysozyme, a hydrolytic enzyme found in the mucus secretion and in tears, is able to cleave the peptidoglycan layer of the bacterial cell wall. Interferon comprises a group of proteins produced by virus-infected cells. Complement is a group of serum proteins that circulate in an inactive state. A variety of specific and non-specific immunologic mechanisms can convert the inactive form of complement proteins into an active state with the ability to damage the membranes of pathogenic organisms, either destroying the pathogens or facilitating their clearance. Phagocytic or endocytic barrier. Another important innate defense mechanism is the ingestion of extracellular particulate material by phagocytosis. In phagocytosis, a cell's plasma membrane expands around the particulate material, which may include whole pathogenic microorganisms, to form large vesicles called phagosomes. Most phagocytosis is conducted by specialized cells such as blood monocytes, neutrophils, tissue macrophages. Inflammatory barrier. Tissue damage caused by a wound or by an invading pathogenic microorganism induce a complex sequence of events collectively known as the inflammatory response. The end result of inflammation is to activate a specific immune response to the invasion or clearance of the invader by components of the innate immune system. The 5 cardinal signs of inflammation are redness, swelling, heat, pain, and loss of function. The cardinal signs of inflammation reflects the 3 major events of an inflammatory response: vasodilation, an increase in the diameter of the blood vessels which is responsible for the tissue redness, and an increase in tissue temperature. An increase in capillary permeability facilitates an influx of fluid and cells from the engorged capillaries into the tissue, resulting in tissue swelling. Influx of phagocytes from the capillaries into the tissue is facilitated by the increased permeability of the capillaries. As phagocytic cells accumulate at the site and begin to phagocytose bacteria, they release lytic enzymes which can damage nearby healthy cells. The accumulation of dead cells, digested material, and fluid forms a substance called pus. Once the inflammatory response has subsided and most of the debris has been cleared away by phagocytic cells, tissue repair and regeneration of new tissue begins. Adaptive immunity. Adaptive immunity is capable of recognizing and selectively eliminating specific foreign microorganisms and molecules, that is, foreign antigens. Adaptive immunity displays 4 major characteristics. Antigen specificity. The antigenic specificity of the immune system permits it to distinguish subtle differences among antigens. Antibodies can distinguish between 2 protein molecules that differ in only a single amino acid. Diversity. The immune system is capable of generating tremendous diversity in its recognition molecule, allowing it to recognize billions of unique structures on foreign antigens. Immunological memory. Once the immune system has recognized and responded to an antigen, it exhibits immunologic memory. That is, a second encounter with the same antigen induces a heightened state of immune reactivity. Because of this attribute, the immune system can confer lifelong immunity to many infectious agents after an initial encounter. Self-non-self recognition. The immune system normally responds only to foreign antigens, indicating that it is capable of self-non-self recognition. The ability of the immune system to distinguish self from non-self and respond only to non-self molecule is essential for the outcome of an inappropriate response to self molecule can be fatal. Adaptive immunity involves 2 major groups of cells, lymphocytes and antigen-presenting cells. Lymphocytes lift the bone marrow, circulate in the blood and lymphatic system, and resides in various lymphoid organs. Because they produce and display antigen-binding cell surface receptors, lymphocytes mediate the defining immunologic attributes of specificity, diversity, memory, and self-non-self recognition. The 2 major populations of lymphocytes are B lymphocytes or B cells and T lymphocytes or T cells. B lymphocytes express a unique antigen binding receptors on its membrane called antibodies. When a naive B cell, one that has not previously encountered antigen, first encounters the antigen that matches its membrane-bound antibody, the binding of the antigen to the antibody causes the cell to divide rapidly. Its progeny differentiate into memory B cells and effector B cells, called plasma cells. Memory B cell has a longer lifespan than naive cells, and they express the same membrane-bound antibody as their parent B cells. Plasma cells produce the antibody in a form that can be secreted and have little or no membrane-bound antibodies. T lymphocytes also arise in the bone marrow and migrate to the thymus gland to mature. During its maturation within the thymus, the T cell comes to express a unique antigen binding molecule called the T cell receptors on its membrane. Unlike membrane-bound antibodies on B cells, which can recognize antigen alone, T cell receptors can recognize only antigen that is bound to cell membrane protein called major histocompatibility complex or MHC molecules. There are two well-defined subpopulations of T cells, T helper or Th and T cytotoxic or Tc cells. T cells display CD4 generally function as T helper cells, whereas those displaying CD8 generally function as Tc cells. After a T helper cell recognizes and interacts with an antigen MHC class II molecule complex, the cell is activated and it becomes an effector cell that secretes various growth factors known collectively as cytokines. The secreted cytokines plays an important role in activating B cells, Tc cells, macrophages, and various other cells that participate in the immune response. Differences in the pattern of cytokines produced by activated T helper cells result in different types of immune response. Antigen presenting cells or APCs are specialized cells which include macrophages, B lymphocytes, and dendritic cells. They are distinguished by two properties. They express class II MHC molecules on their membrane, and two, they are able to deliver a co-stimulatory signal that is necessary for T helper cell activation. Antigen presenting cells first internalize antigens either by phagocytosis or by endocytosis and then display a part of that antigen on their membrane bound to an class II MHC molecule. The T helper cells recognize and interact with the antigen class II MHC molecule complex on the membrane of the antigen presenting cells. An additional co-stimulatory signal is then produced by the antigen presenting cells, leading to activation of the T helper cells. Antigen selection of lymphocytes causes clonal expansion. Antigen selection of lymphocytes causes clonal expansion. A mature immune-competent animal contains a large number of antigen-reactive clones of T and B lymphocytes. The antigenic specificity of each of these clones is determined by the specificity of the antigen binding receptor on the membrane of the clone's lymphocytes. The specificity of each T and B lymphocyte is determined before its contact with antigen by random gene rearrangement during maturation in the thymus or bone marrow. In clonal selection, an antigen binds to a particular T or B cell and stimulates it to divide repeatedly into a clone of cells with the same antigenic specificity as the original parent cell. Specificity is shown here because only lymphocytes whose receptors are specific for a given epitope on an antigen will be clonally expanded. Self-non-self discrimination is accomplished by the elimination during development of lymphocytes bearing self-reactive receptors or by the functional suppression of these cells in others. Immunologic memory also is a consequence of clonal selection. During clonal selection, the memory cells which have a longer lifespan than the naive lymphocytes from which they arise are also produced. The initial encounter of a naive immunocompetent lymphocyte with an antigen induces a primary response. A later contact of the host with antigen will induce a more rapid and heightened secondary response. The amplified population of a memory cells accounts for the rapidity and intensity that distinguishes a secondary response from the primary response. Conclusion. In conclusion, I would like to state that the two major components of the immune system are interdependent on each other. The innate immune system gives the first line of defense to protect against any pathogen that enters our body. When the pathogen can overcome the innate immune system, the second line of defense, that is the adaptive immune system, comes into play. Many cells involved in the innate immunity produces certain cytokines that attract lymphocytes, which are the major cells in the adaptive immunity. The adaptive immune system gives a more specific and vigorous immune response to completely eliminate the pathogens from our body.